So to start off, we'll talk about Illumina TruSeq RNA Access Library Prep Kit and how we overcame some challenges associated with the workflow on our Biomek FXP dual hybrid platform. I don't have slides of the system, so if you can recall the instrument that Zach had shared with you earlier, this application and the next one were both developed on our hybrid dual arm system, so it deploys a span 8 pod as well as a multi-channel 96 well head for combination of efficiency and accurate enzyme transfers. So benefits of the RNA access chemistry. It's designed to address challenging RNA-seq and target capture workflows when using degraded and FFPE or formalin-fixed paraffin-embedded derived RNA. The challenges of FFPE are gene expression profiling are overcome by combining RNA-seq library preparation with an exome enrichment. The exome enrichment focuses on the coding region of RNA, so you're going to require less sample input in your library prep and sequencing reaction. So we're able to get successful libraries generated from fresh frozen tissue at 10 nanograms of input, and for FFPE material starting at 20 nanograms for degraded FFPE samples. So as a result of the exome capture, the sequencing is going to require fewer reads, which will allow you to increase the amount of samples pooled in your sequencing reaction and therefore provide more cost-effective transcriptome analysis. So this slide diagrams the Illumina manual workflow for the RNA Access Kit. If anyone's familiar with it, it's a monster. Zach's HLA method that he described was big in of it itself, but the RNA Access Kit is equally big. So it's multi-stage process, including fragmentation, cDNA synthesis, first strand and second strand synthesis, typical ligation, 3' adenylation and ligation steps. There's an enrichment following all that, and those steps are the pre-hybridization component of the kit. Between the pre-hybridization component and the hybridization capture, there's quantitation steps and some normalization and pooling that are required going into the capture. So Illumina's chemistry is set up, or the kit is filled such that you can get 48 libraries generated out of a kit. That's pre-capture, and then they provide enough reagents on the hybridization capture to perform 12 4-plex capture pools. So our method is going to be designed at around roughly a 2 kit per run throughput. So following the pre-capture protocol, we have 2 rounds of hybridization capture and cleanup, followed by another enrichment of the capture product. So in the course of all that, you can see that there's quite a lot of steps of enzyme additions. There's 6 total Ampere cleanup steps associated with the full process, as well as the bead-based hybridization capture, which is similar to a cleanup protocol with a lot of liquid transfers. So challenges to the workflow. I touched on the amount of time-consuming and highly complex protocol steps that are involved. So we have 9 reaction mix additions. There's a total of 6 Ampere cleanups and 2 hybridization captures. The low input sample requirements are a challenge to the workflow, so accurate pipetting, reagent delivery, balanced chemistry is important to ensure that we're getting the most robust results from the kit. The kit can handle degraded samples from FFPE, and so again, we have to be careful in the way we're processing these, that they're treated with precision and accuracy. So the manual protocol, according to Illumina, takes 11 hours of hands-on time to generate 17 to 48 libraries and 3 full days to complete. So the 3 full days is 24 hours, but we know that technicians or scientists in the lab aren't working at the bench for 8 solid hours every day, and work tends to revolve around your schedule with meetings and other such things. So typically the workflow, from what I've seen, is more on the lines of 4, maybe 5 days, depending on how each of the steps break down. So if you're willing to stay 10 hours on day 1, you can maybe knock it out in 3 days, but that's kind of a stretch. So there's hundreds of pipetting steps involved with the process, and the more samples you're processing per run, it's increasingly difficult to get that work done manually, especially with talking about Ampere bead addition and cleanups, et cetera. So we decided to automate it on the FXP platform. It's the perfect system for this workflow. It needs large deck capacity, and it has the dual pod system for enzyme delivery and precision pipetting, as well as the multi-channel pod that creates efficiency around our Ampere cleanups, and there being 6 of them again in this run. So we fully automated the highly repetitive and time-consuming tasks, again, all those mix additions and cleanups, and the full protocol can be automated in approximately 20 hours of instrument runtime, regardless of sample number. So that's the benefit of the automation, is as you increase your sample number, your time for processing doesn't change as significantly as it does when you're performing it manually. So each Axis kit has 48 pre-capture libraries that you can generate. Our system is capable of processing up to 96, so that'll be a two-kit run on the pre-hyb portion of the protocol. On the hybridization protocol, we have it set up to process 48 4-plex capture pools, which is actually 4 full RNA Axis kits. So depending on how you structure your workflow, 2 rounds of library prep can be done, and then 1 round of the hybridization capture protocol. And the net result would be a total of 192 hybridization capture pools at the end. So there's different ways to structure your workflow. That's quite a lot of samples, and the system's fully capable of handling that. With that being said, all that work only requires 1.25 hours of on-time or hands-on handling, as compared to 11 solid hours of manual sample manipulation from the manual protocol. So there's a clear reduction in hands-on time with this method. This is a slide showing the automated workflow. It's a little bit difficult to see, but each of those purple boxes represents a module. The first 2 and the last 2 are part of the Illumina protocol. The center one is a quantitation module that we added in to allow picogreen quantitation of your pre-hybridization samples, and it also accommodates normalization of those samples and pooling going into the hybridization protocol. So each of the workflows, each of the modules follows the vertical workflow, and each of those can be started and stopped independent of each other to create some flexibility around how your lab work is scheduled. In addition, if there is a safe stopping point throughout one of those modules, then we've built a stop and start point into the method at that point. So the green arrow represents an alternate stop point, and the little yellow, it's a clock if you can't see it. Those are alternate stop points, and that's the point at which you can take your samples off the automation, store them, and then come back on the next day or later in the day to continue the process. Okay, to facilitate execution of the method, all of the feature-rich methods that we deliver from Beck & Coulter are going to contain a user interface, and that user interface allows the user to select the portion of the method they want to run in all different types of method parameter options. So in this case, the user can choose anywhere between 1 and 96 samples, choose one of the 5 distinct modules, and if there's built-in stop and start points within each of those modules, they can also choose which portions of that module they want to run, one or the other or both. There's flexible index, labware, and transfer options. So the RNA Axis kit comes with indices and tubes versus some of the pre-arrayed plates. There's options to use either the manufacturer's source tube or to array your indices into your own custom labware. And then with that being said, we have standard transfer protocols, and then we have custom transfer protocols. So we have an A1 to A1 transfer, just a standard straight well mapping from the index plate to the sample plate. And we also have the ability to take directly from the source tube with a custom file that allows any index to be placed in any well. Again, we have the quantitation setup module included since it's required to continue on to hybridization, and that's going to perform a picogreen quantitation setup along with normalization and pooling for the 4-plex pool hybrid samples. And lastly, we have the option to perform all incubations and thermocycling on an integrated T-robot thermocycler. And that's going to be the true power of the system when you can have a complete walk-away capability, start your method, let it run for 4 or 5 hours or whatever length of time that module takes, and come back and your samples will be ready to go for you. Okay, after the user starts the run, based on the sample number that they have chosen, they're going to be presented with a reagent calculator. This is another feature that's in all of our methods. And what it does is it provides master mix recipe preparation instructions to the user. So this is going to prevent errors from users having to calculate how much of component A and component B to put into master mix 1. It's going to give you that information based on your sample number. It includes excess volume for pipetting and for dead volume in your reagent tube. After the master mixes are prepared, it'll give detailed labware configuration, sample and reagent tube and reservoir fill volume. So it'll tell you how much of your master mix to place in what tube, in what piece of labware. And then that'll be placed on the deck and instructed through the instrument setup step. And again, it automatically updates based on sample number and method options selected. So it's going to be a dynamic reagent calculator. The user doesn't have to think about what volumes they need based on their options. The system's going to do that for them. Okay, so we collaborated with Illumina on an experiment set, taking UHR, human reference RNA, 2 pools of that, along with human brain RNA and some FFPE control, and put it through the full protocol. And the top left slide there just shows the reproducibility of the library insert size. And the bottom slide is going to show the average yields obtained from each of those 6 hybridization captured pools. So the first 4, the first 2 are the human reference RNA, the middle 2 are human brain control, and then the last 2 are the FFPE samples. So all samples generated greater than 75 nanomolar concentrations, and including the FFPE and non-FFPE samples that were less degraded are generating yields of greater than 100 nanomolar. So again, it is able to generate robust, high-yield libraries. So in collaboration with Illumina, we generated sequencing data. They had samples sent over to them, ran them on their 2500 sequencer, and then analyzed via base space. And sequencing metrics are as follows. So the top left slide is the alignment distribution, and it shows a high % alignment to the coding region, less than 3% abundance, and very low ribosomal RNA component. There's an average of greater than 12 million pass filter reads per pool, and the results are highly reproducible across sample types, including the low quality FFPE material. The bottom graphic indicates % duplicates. So the first 4 boxes are going to be the non-FFPE samples, showing very low % duplicates of 20% or less. And even the FFPE degraded material is showing a relatively low % duplicate of approximately 35%. And across the board, the median insert size is highly reproducible across all sample types, and that will be the small triangles there across the center of the graph. Okay, so in summary, our method is able to generate 96 highly enriched exome libraries, which will turn into 24 4-plex capture pools in less than 3 days with only approximately 1.5 hours of hands-on time. It includes a feature-rich user interface and a reagent calculator to create flexible workflows and reduce setup errors. And then as far as sequence data is concerned, we have greater than 12 million pass filter reads with minimal % abundance and % ribosomal RNA component with highly reproducible results.